Right. Let's jump straight into this explainer. Imagine a patient hits the ER with a severe, life-threatening bacterial infection, like sepsis. What's happening in that moment is literally an invisible war raging inside them, and the stakes couldn't possibly be higher. Every single hour that ticks by without the right treatment drastically spikes the mortality rate. So to win this war before time runs out, medical professionals basically have to act like high-stakes detectives. Today, we're going to walk through the complete diagnostic algorithm for bacterial infections. We'll track the exact step-by-step journey a sample takes from the patient's bedside all the way to finding the perfect targeted antibiotic to take the culprit down. To break this down, we're treating the clinical microbiology lab like a crime scene investigation. We'll be moving through these 5 critical phases, starting with the blueprint, then securing the scene, moving to the stakeout, finding the weapon, and wrapping up with the fast track.
All right, starting with part 1, the blueprint. This is the roadmap of our microbe hunt. Here is the master plan that every single diagnostic algorithm follows. It always kicks off by securing the biological evidence, getting that perfect specimen from the patient. Next up is direct detection under a microscope to grab a quick initial profile of the suspect. Then we culture and fully identify the bug, effectively pulling it out of hiding. Finally, we run antimicrobial susceptibility testing to lock in our ultimate weapon. And this order? It's strictly non-negotiable. You can't exactly test a weapon on a suspect you haven't even caught yet, right? Though, as we'll see in step 5, modern rapid diagnostics are totally flipping the script on this timeline.
Moving to phase 2, securing the scene. This is all about quick clues and early action, because once we've collected that specimen, be it blood, sputum, or spinal fluid, we are officially on the clock. Here's the thing. Bacteria multiply like crazy, so doctors literally cannot wait days for lab results to come back. They have to launch what's called empirical treatment immediately. Think of it like using a massive wide net or a shotgun approach to just hold the line, because we don't have the intel for a sniper rifle yet. It's a highly educated guess based on rapid testing in the patient's symptoms, allowing doctors to deploy broad-spectrum antibiotics to fight off the most likely suspects while the lab does the heavy lifting behind the scenes. And within minutes, a super simple direct detection method, like a gram stain, can give us a massive break in the case. Notice those dark purple spherical clusters right there? Those are gram-positive cocci. Just by looking at their shape, how they group together, and how they absorb that stain, lab techs can instantly eliminate thousands of other potential suspects. It's an immediate microscopic profile that gives the medical team their very first actionable clue.
So why is this rapid profiling absolutely crucial? Well, for fastidious organisms, which are essentially the super picky eaters of the microbial world that take forever to grow in a lab, or for terrifyingly fast infections like pyogenic meningitis, this early clue is a total game changer. It points the doctors in the right direction to administer life-saving empirical antibiotics way before the multi-day culture even finishes.
Okay, part 3, the stakeout. This is where we slow things down a bit to actually grow and rigorously interrogate the bacteria. This is the classic microbiology you're probably picturing. By cultivating the bacteria on different nutrient plates, or agars, we literally force them to reveal their identity. Check out the striking green colonies of Pseudomonas on the left, completely distinct from the bright pink lactose-fermenting ones on the right. By giving them different environments, microbiologists watch how they behave. Just their color, shape, and growth patterns on these specific gels can narrow the suspect list from a giant family down to a specific genus.
Once we've got a solid visible colony, it's interrogation time. We run the suspect through a gauntlet of biochemical tests, like indole, citrate, urease, and TSI, to grab its exact metabolic fingerprint. We're checking for survival enzymes like catalase and oxidase. Basically, we're asking, how does this bug breathe? What sugar does it ferment? This deep dive fingerprinting lets us perfectly identify the exact species. We finally have a confirmed name for our culprit.
All right, on to part 4. The weapon. Identifying the bug is huge, but it's really only half the battle. Now we actually have to find out what will kill it.
This right here is the holy grail of the entire diagnostic algorithm. We are transitioning the patient from that initial broad-spectrum educated guess, the empirical therapy, straight into pathogen-directed therapy. Once we know exactly what the organism is and what it hates, we can finally swap out the shotgun for a sniper rifle. We drop in a narrow-spectrum antibiotic that acts like a heat-seeking missile, destroying the infection while leaving the patient's healthy microbiome completely alone.
To figure out the exact dosage, we use antimicrobial susceptibility testing, or AST. Take a look at the series of tubes on your screen. We expose the isolated bacteria to weaker and weaker concentrations of an antibiotic. The cloudy tubes? That means the bacteria are laughing at the drug and actively growing.
But right at that 8 milligrams per milliliter mark, the tube is crystal clear. That is our minimum inhibitory concentration, or MIC. It's the absolute lowest dose required to stop this pathogen dead in its tracks.
Now, this susceptibility testing is obviously vital for the individual patient survival, for sure. But it also has a massive global impact. Tailoring treatment allows doctors to practice critical antimicrobial stewardship. It prevents us from just spamming broad-spectrum drugs, which, as we know, is the main driver of global antibiotic resistance. By using exactly the right weapon, we protect our strongest antibiotics for the future. Which brings us to our final phase, Part 5, the fast track. Because honestly, the traditional algorithm works brilliantly, but it takes days. What if we could bypass that slow stakeout entirely?
Enter the molecular revolution. Advanced rapid diagnostics are totally rewiring our standard lab playbook. Instead of waiting days for bacteria to grow on a plate, we just look directly for their DNA or RNA in the original sample. Using automated multiplex PCR, which amplifies the genetic code, or mass spectrometry tech like MALDI-TOF, we are crushing that multi-day wait down to mere hours. We're finding the exact genetic needle in the haystack without having to actually grow the haystack first.
Just 1 hour. That is literally all it takes for an automated PCR system to screen a patient's spinal fluid for 14 different common meningitis pathogens simultaneously. 1 single hour to identify the exact DNA of a life-threatening brain infection. This kind of speed is completely unprecedented, and it fundamentally changes how fast we can pivot from a broad empirical guess to a precise, targeted strike. It's saving countless lives. So as we look ahead to the future of the medical lab, it leaves us with a pretty fascinating question to chew on. With molecular profiling getting faster, cheaper, and more comprehensive every single year, how long until our classic step-by-step culture plates and test tubes become a thing of the past? We are quite literally watching this algorithm evolve in real time. Thanks so much for joining me on this explainer, and remember, always keep questioning how the invisible world around us is decoded.